To be from Rhythm Pharmaceuticals, and I'd like to welcome Lex van der Ploeg to the Boulder Peptide Society. Thank you, Lex.
Hi, thanks. Good morning. Thanks for the invitation to the organizers to be able to present the work that is ongoing at Rhythm Pharmaceuticals. Really enjoyed this morning's session. I will take you through the background and development of Setmelanotide, a small octapeptide applied for the treatment of rare monogenic forms of obesity. Rhythm is a company with two compounds in development. Can you see the pointer? Well, there it is. It's very faint. Okay. We have a ghrelin agonist RM-131, which is a small pentapeptide in development for the treatment of diabetic gastroparesis. And we'll be talking about the MC4R agonist peptide RM-493. We have a small management team. We're a virtual company. Everything works through CROs and contract laboratories, and has been quite effective since the initiation of the company. So RM-493, known as Setmelanotide, is a peptide for reducing weight and insulin resistance. We're treating rare genetic forms of severe obesity, and I will explain to you the work that we've done initially in general forms of obesity, and then within the pathway, which I will highlight with regards to the individual genetic forms that we're targeting. Our target product profile here is double-digit weight loss, over 10%. And that is through the control of appetite and increased energy expenditure. And I'll provide the data for that explains that we are indeed meeting both targets. The outcomes should be improved glycemic control as one of the core comorbidities, and improved cardiovascular health. When you look at the major regulatory factors that are impacted by the synthetic melanocortins, they really come down to those that are in the orexigenic path and in the anorexigenic path. From an endocrine perspective, the hormone ghrelin, discovered in 1999, is well known with regards to its impact on metabolism, and of course anorexigenic leptin out of 1996 events. Neurotransmitter NPY, neuropeptide, very important in appetite control, and an antagonist of the melanocortinergic system, AGRP. And of course MSH as the major driver of the melanocortinergic pathway, where RM-493, Setmelanotide, is a mimetic of the active entity MSH. Now, to give you a little bit of background on the neurological pathways in the hypothalamus, core component is there are two parallel pathways. Leptin as well as ghrelin interact on these, and in parallel there is control of NPY and AGRP through a variety of different hypothalamic nuclei, which all lead to the increase in food intake due to the action of ghrelin. Leptin, of course, suppresses this. And then leptin's anorexigenic effects on CART POMC neurons, again, through a range of different hypothalamic nuclei that can decrease food intake and increase energy expenditure. And it is the interplay between these symptoms that the melanocortinergic peptides interact with and can drive and help control. Now, the potential genetic targets that Setmelanotide activates are located in the POMC pathway, and I'll speak to a little bit of background there with regards to what that entails. Pro-opiomelanocortins, in combination with a number of different peptides that cleave this prohormone, lead to the production of MSH, that of course then hits the melanocortinergic receptors, G-protein coupled receptors on MC4 neurons, and thereby have a direct impact on appetite and weight. This is a very simplified model where you have a linear outline, satiety signals, for instance, leptin coming in through the leptin receptor, activating POMC neurons, leading to the production and release of the peptide, PCSK, one of the hormones that cleaves it, MSH production. Genetic deficiency can target several targets related to this pathway, and it can result in severe obesity. For instance, there are currently about 50 patients known worldwide, the population is actually larger now, 120, 150, that are POMC deficient and or PCSK1 deficient. And the epidemiology of these is not well characterized, so it's really early days. Of course, there's 100 patients known at the moment, while the epidemiology forecasts larger populations. This is an ultra-orphan, and we're looking at this pathway with regards to where the patient populations are located so that we can target the largest possible population within this pathway, and we believe there are many, many thousands. There are other components out of the pathway as well, and they include the Prader-Willi syndrome. And new evidence recently released a couple of years ago highlighted that the MAGEL2 gene, which is also located on chromosome 15 and inactivated in this imprinting disorder, impacts directly on the functionality of POMC neurons. And there are about 9,000 patients in the US that are Prader-Willi syndrome characterized. Patients that are defective for the MAGEL2 gene alone develop a Prader-Willi syndrome-like clinical presentation, where many of the aspects of the Prader-Willi syndrome are there, but not all. And it is a, let's say, a heterogeneous penetrance of that null phenotype in that population. MAGEL2 null mice that have been evaluated, which are a model for the Prader-Willi syndrome, have defective POMC neurons, and hence this is a defect that is located upstream of the MC4 receptor as well. And the model, therefore, is that as a result of this deficiency, MSH is not properly generated, and hence such upstream defective individuals should be hyper-responsive to compounds like setmelanotide. And the model that we've therefore pursued is that when you compare the activity of wild-type obese patients to patients that, for instance, have a defective MC4 receptor, the outline would be if you have fewer receptors, you will be responsive by use of setmelanotide, but not as responsive as when you're wild-type. And of course, if you don't have MSH and you can supplant the hormone with application of RM-493 or setmelanotide, you anticipate to get a significant response because you're replacing a hormone that was absent due to that particular genetic defect. Now, just a bit of background on the G-protein coupled receptor agonist neuropeptide pro-opiomelanocortin. It generates, relevant for this presentation, gamma MSH, alpha MSH, and beta MSH, as well as beta endorphin. They are cleaved out of this pre-pro protein. And these ligands have activities at the MC3 receptor and the MC4 receptor, and both receptors are involved in functioning of energy expenditure as well as feeding behavior. I earlier highlighted that this system is rather complex. There is an antagonist at the MC3/4 receptor, AGRP. It is located uniquely at the arcuate nucleus and thereby signals through the hypothalamus and impacts on the neurons that I've just highlighted that are regulated by the agonist alpha MSH, and of course when you apply setmelanotide by that particular ligand. What is important is that a lot of studies have been conducted over the last couple of years to define how much brain penetration is needed to attain efficacy with melanocortinergic agonists. And it's easiest to think of this as a two-compartment model. When you get into the brain, ICV, you definitely impact food intake and energy expenditure. But if you take peptides that are not brain penetrant or compounds that are not brain penetrant, or at least you can measure them by analytical means in the brain, you can still get equally effective modulation of appetite and energy expenditure. This is an experiment that kind of like highlights what may be happening there. These are coronal brain sections of the rodent where we look at the binding of iodinated MT2, thereby identifying the distribution, median eminence, ventral medial hypothalamus, of those neurons where MC3 and MC4 receptors are located in the rodent brain. What we did subsequently is we first used cold, and I'm not showing you that data, but an iodinated version of MT2 with no radioactive label onto it, and we determined that that peptide is equally potent in suppression appetite and changing energy expenditure as wild-type MT2. What we subsequently did is we determined in giving this peptide IV to the rodent and then subsequently washing out the unbound label, taking the brain, sectioning it, and taking the coronal brain sections and putting them up for autoradiography where it binds. How much does that iodinated MT2 peptide actually penetrate into the brain. And surprisingly, what we were able to show is that we cannot, either by mass spec or by other means, detect it at deeper brain sites, but it uniquely binds to the circumventricular organs, those areas where the brain has its neurons sticking outside of the blood-brain barrier and samples activities in the plasma, the median eminence, the choroid plexus, and here one of the subfornical organs. And hence what it highlights is that at least for peptides, where you cannot measure overt brain penetration, you can get good activity on the suppression of appetite and energy expenditure. Doesn't mean it doesn't get into the brain, just means we can't measure it yet, and our methods are probably not elegant enough, but there are receptors there at the circumventricular organs outside of the blood-brain barrier that are the recipients of signals, and we anticipate thereby signal into the brain and can change appetite and energy expenditure. What is also important is that with regards to the activity of melanocortinergic peptides and those neuropeptides that stimulate appetite and reduce energy expenditure, that NPY being one example, that you look here at satiated rats that are either ICV getting NPY or bovine pancreatic polypeptide hitting the Y4, Y5, and the Y1, Y2, and Y5, and they are then administered MT2 ICV, that you can see that in these rodents, compared to control, there is a significant suppression of appetite by activation of melanocortinergic pathways. That means melanocortinergic agonism can overcome NPY-induced food intake. It's important because these are dominant players that control appetite and energy expenditure. Subsequently, we looked at the effects of ghrelin as well, and again look at the dependence of ghrelin on NPY and melanocortinergic pathways, and these are rodent studies where we looked at knockout rodents. This is AGRP wild type and NPY wild type. This is vehicle, this is a ghrelin agonist where you stimulate appetite significantly. You can then see if you knock out the NPY peptide, you have a significant reduction in the activity of the ghrelin agonist. And if you knock out both IGRP and NPY, you virtually have no activity. So both the activities of the ghrelin system work through AGRP and NPY, as well as that the melanocortinergic peptides can impair on the NPY and of course the AGRP activity, because it's simply an antagonist of the melanocortinergic receptors. So in summary, what that means is setmelanotide is a potent melanocortin agonist, and it impacts several orexigenic pathways. Ghrelin, NPY, and AGRP-stimulated food intake are anticipated to be suppressed by the application of melanocortinergic agonist. There are other interactions with endocannabinoids, histamine, muscarinics that we're not going to be discussing here, but we anticipate, therefore, that RM-493 as a melanocortinergic agonist has a profound impact on metabolism because it hits the core pathways that are important for the control of energy expenditure and appetite. Now, setmelanotide, its selectivity is given here out of an EC50 assay measuring its GPCR activation through cyclic AMP. It hits the MC1 receptor with about a 30-fold selectivity over MC4. It doesn't see the MC2 receptor, which activates corticotroph ACTH responses. It sees MC3 again with the same selectivity, potent at MC4, and it doesn't significantly see the MC5 receptor. And it is this peptide that we've taken forward in individual animal models and through safety toxicology into clinical trials, and I'll show you that data in the coming couple of slides. An initial outline is that setmelanotide was evaluated in 12 obese, insulin-resistant monkeys, where the peptide was infused over a period of 8 weeks. The study endpoints were food intake, fat mass, insulin sensitivity, monitoring cardiovascular parameters, lipids, activity and behavior, and energy expenditure. And these were really important and determining studies because in the past, all efforts for the development of melanocortinergic agonists have failed because of an impact on increase in heart rate and increase in blood pressure, systolic and diastolic. And that earlier had led to the demise of any of these other compounds, which were all small molecules largely and non-peptides. So this was a determining experiment for us because we needed to determine first efficacy, what does it do on body weight, energy expenditure and appetite. Secondarily, is it cardiovascularly safe? Is it different from compounds that earlier had significant cardiovascular effects? What you can see here, out of the 12 monkeys individually numbered, and in the gray zone you can see the duration of the infusion of setmelanotide, is that over the 8-week period in these diet-induced obese primates, there was about a 13% reduction in body weight, mainly as fat mass. In addition, what we were able to show that, interestingly, during the washout period, you can see that in most, for a period of 2 to 6 weeks after we had stopped the treatment with setmelanotide, there was a sustained, steady body weight and a lack of an immediate increase or an immediate rebound. We do not understand the reasons for that, but it's intriguing because it indicates that there has been a longer-term effect that is not directly dependent on the pharmacological activation of the MC4 receptors that setmelanotide targets. In addition, what we were able to show, and I'll show you some examples of that, with continuous telemetry, there were no cardiovascular responses on heart rate, systolic or diastolic blood pressure. Appetite was significantly reduced over the first 4 weeks and then leveled out and tended to come back a little bit over the next 4 weeks, but stayed below normal. And of course, body weight continued to go down. There were effects on insulin and glucose, and major other parameters were non-impactful with regards to safety measures that one would like to look at. This is an example of a head-to-head comparison in obese primates, where after this study, we did a crossover study in telemetried rhesus monkeys, and thereby defined, if we went in with the Lilly compound, LY211268, which failed in the clinic as a result of significant cardiovascular effects, took these animals on drug, put them on the Lilly compound for several days, took them off, washed them out, and then put the same animals back on setmelanotide to determine whether there was a differential effect on the heart rate and blood pressure. I'm only showing the systolic blood pressure here, vast increases in blood pressure in this particular animal, this is the same animal shown here left and right, and this is continuous telemetry, and this is the setmelanotide treated monkey which had no cardiovascular effects. The implant failed here, but we have many, many more, but I'm showing you this one as the Lilly effect here was quite profound. And this is another example of a much less drastic increase in this particular animal, where you can see a significant increase as a result of the administration of the Lilly peptide, and no significant effects of setmelanotide on the cardiovascular parameters. So at the end of this study, we walked away with a peptide that impacted appetite, impacted energy expenditure out of doubly labeled water, which was up by about 12–13%, which was cardiovascularly clean in the continuous telemetry animals over the 8 weeks of treatment and drug treatment in the diet-induced obese weight loss that you saw. And here in the crossover study, where you can see that there's a clear discrimination to what the Lilly peptide brings about and what setmelanotide brings about. The peptide has subsequently moved forward through safety toxicology studies and is in clinical trials, where I'll show you the data out of regular severely obese individuals, out of MC4 heterozygous patients that have one defective receptor, and then within the pathway, we've worked on looking in the pathway at defects that are upstream of the MC4 receptor, where I'll show you examples of patients that are POMC deficient, that have null deletions by different mutations, and thereby cannot make gamma, alpha, or beta MSH and are fully defective. So, of course, critical safety parameters that were evaluated are heart rate, blood pressure. We look at skin color, because we do affect the MC1 receptor potentially, and penile erections and sexual activity, as it's been documented in the past, that the MC4 receptor can give rise to changes in sexual behavior. Multiple ascending dose study, I will highlight to you, initially was done 24-hour infusion in several cohorts at 0.01 mg/kg for 14 days. 0.01 mg/kg for 28 days, 0.015 mg/kg for 28 days. We also did sub-Q injections, and we're now dealing with a once-a-day injection. This was done twice a day at 0.075 mg/kg. And of course, safety, ambulatory blood pressure was measured throughout, weight loss, food intake, PK, and maximeter for skin color. So a careful evaluation, and we have thus far evaluated about 200 patients with setmelanotide in this setting. And this gives you a summary of those results, where it is best to first look at the 4-week treatment here in wild-type obese individuals, where over a period of 4 weeks, you can see we had roughly a 4 kilogram weight loss. I'd say about 0.9 to 1 kilogram of weight loss per week. But you can see there's some variability at this dose here, 0.015 mg per kg again infused. The weight loss was more like 0.6 kilograms per week. Over a shorter duration treatment, 2 weeks, about 0.8 kilograms per week, not dissimilar from the activity seen here at 0.01 mg per kg, and with a BID injection over the 2-week period, roughly 1.4 kilogram weight loss per week. So this highlights that in this setting, and we had significant weight loss as a result of the compound either being infused or being administered by BID sub-Q administration. And there were no noted significant safety issues, cardiovascularly clean, no measurable cardiovascular effects as a result of the ambulatory blood pressure measurements. What we did note is slight increases in skin color as a result of hitting the MC1 and hence a tanning that is equivalent to what is observed with a regular suntan. It plateaus after about 2 weeks of drug treatment. Interestingly, we also treated patients that were heterozygous for defects in the MC4 receptor, which means one of the receptors is wild type and the other receptor has a mutation. And in this group, there were several patients that had a complete loss of function at their mutated receptor, and others had partial loss of function at their mutated receptor. You can see that these also respond quite well over the 4-week period with roughly, let's say, 0.6 to 0.7 kilogram per week weight loss. So we can show that wild types and heterozygous deficient patients can benefit from treatment, and we can indeed activate the receptor in these individuals productively and thereby overcome their defect that is causative for their obesity. That is, of course, attractive, because if you look at patient populations where you want to make sure that they will respond, being dependent on a defect in the receptor for your obesity gives you a high likelihood of treating a effective responder population. We also did studies collaboratively with the NIH looking at energy expenditure together with the NIDDK, where patients were in metabolic chamber for a period of 3 days and were at the end, at the 3rd day of treatment, as a result of the infusion of the compound, over a period of 30 minutes per time period, evaluated for resting energy expenditure. And here RM-493, placebo, percent change, and the p-value is highlighted, where we look here at resting energy expenditure, the most important component, where we had a 6.85% increase in this study where patients were treated and measured over a period of 3 days. There were effects on appetite, energy expenditure was increased, and such an effect on energy expenditure, if you were simply to calculate what its impact would be over a period of roughly a year, would already give a 7 kilogram weight loss over that time period. Again, no effects on cardiovascular parameters that were carefully monitored, and no safety effects noted in this study. Of course, what one has to keep in mind is that generally on weight loss there are compensatory mechanisms that can reduce energy expenditure, and hence this increase is relevant, because even staying flat and not compensating for the weight loss by a decrease in energy expenditure can have a significant impact on the long-term outcome of such weight loss studies. Now, here the mean change from baseline with regards to systolic, diastolic blood pressure and heart rate, and this is from baseline where there were no significant effects noted in the trials that I showed you earlier, and the 34 for RM-493 and 21 for placebo. And the differences from placebo are listed here. Again, no noticeable cardiovascular effects, and hence a compound that is quite different from what has been noted earlier with compounds in development from a diverse range of companies. Now, like I indicated earlier, we know that we're dealing with a pathway. There are effects upstream of the melanocortin-4 receptor and effects at the melanocortin-4 receptor that we can anticipate RM-493 to overcome. It is known that leptin-deficient mice are very sensitive to the treatment with MT2, actually more sensitive to treatment with MT2 than their wild-type counterpart. We have subsequently looked at leptin receptor deficient mice and rats, and comparably these are hypersensitive, more sensitive than wild-type rodents to treatment with setmelanotide. And I'll show you the data. Magle-2 null mice, again, hypersensitive to treatment with setmelanotide, again, as a result of replacing the lack of the endogenous hormone MSH. POMC null mice have been documented to be sensitive to MT2, and of course we've shown that MC4 knockout mice are fully resistant to the setmelanotide effects, and I'm not going to show you the data, but wild-type and heterozygous deficient MC4 receptor mice, that the wild-type mice are more sensitive to treatment than the MC4 heterozygous rodents. What that speaks to is that there is a pathway where we have upstream of the melanocortinergic receptor a number of defects. leptin receptor, POMC, PCSK, and MAGEL2. There are other genes that we have not yet evaluated, serotonergic, nuclear receptor, carboxypeptidase E, that are predicted all to predispose to hypersensitivity to replacement therapy with setmelanotide. The downstream, we have shown sensitivity here, but not as good we anticipate as the wild type. And there are a number of genetic defects that are predicted to be fully resistant. This we've shown in rodent. And then there is a large array of at least 20 genes that are currently unknown, but we believe will fit the pathway. And we have to define which patients are going to be responsive and which patients will not be responsive. And we're in the process of doing that. So within this pathway, we're now going to look upstream because we have good responses, 0.9 kicks per week of weight loss in wild-type patients. We want to know whether we can do a lot better with RM-493 and other patient populations. This is the rodent study I highlighted, MAGEL2 deficient rodents, where you see a 3-hour time period here, where you see MAGEL2 deficient rodents over this period treated with RM-493. This is the wild-type treated with RM-493, and this is the vehicle in black and the MAGEL2 vehicle here in red. You can see an absolute very large difference. There's essentially no response on the wild type and a significant suppression of appetite in the MAGEL2 null. As this highlights that rodents that lack endogenous ligand perform better on supplanting setmelanotide than those that are wild type. A similar effect was seen for DB/DB mice. Again, here's the DB/DB rodent, and here are the wild types and the vehicle-treated rodents. A large effects where this animal, the group of animals that is defective in the leptin receptor, is hyper-responsive. So we're looking upstream of the melanocortinergic receptor. Recently published in the New England Journal of Medicine by Widom and the Charité Hospital with Peter Koonen, where we treated patients that have POMC deficiency. And POMC deficiency obesity is characterized by severe early onset obesity and severe hyperphagia. Additional characteristics that can include red hair and light skin as a result of the lack of melanocortin signaling, ACTH deficiency, which needs to be treated, otherwise lethal, and often siblings in families where there is a pedigree, if it's not early recognized, will not live beyond 1 year of age. Early deaths of siblings, therefore. Hypogonadotropic hypogonadism, also known within the gonadotropin POMC pathway. PCSK1 deficiency fits this model as well, with a history of diarrhea and malabsorption during infancy, followed by severe obesity. Now, the 2 patients for which I will show you data are highlighted here. Patient 1, at the time of treatment, they were both adults over 21 years of age, had significant weight gain throughout her life, ending up here at about 18 years of age with a BMI of about 50. So she is severely, morbidly obese. All efforts during her life to try and prevent the weight gain were short-lived and not successful. Patient 2, a similar paradigm with regards to continuous weight gain and a weight of about 150 to 160 kilograms at about 18 or 19 years of age. And you can see here the percentiles for other non-genetically defined individuals and those that fit the curve with regards to being at 50 percentile. So these patients suffer from unrelenting appetite, continuous drive for food intake, thereby morbid severe obesity, insulin resistance, significant impairment of their cardiovascular parameters. And we've learned as a result of treatment of these patients what the benefit is of setmelanotide treatment over a period of time. I'll show you the 2 patients in the next couple of slides. How am I doing for time? 5 minutes? Okay. POMC deficiency in a phase 2 study in patient 1. And what you can see here is over a period of about 86 weeks, this patient lost about 60.5 kilograms of body weight. During the initial phase, she was escalated from 0.25 milligrams to a half milligram to 1 milligram to 1.5 milligram sub-Q injected once per day. Her weight loss over the first 13 weeks was about 2 kilograms per week, roughly, as you can see here. At that point, she was taken off drug. As a result of the drug holiday she was in, her weight immediately increased, her appetite immediately increased, and she was severely distraught by those changes as a result of the lack of therapy, and was subsequently put on therapy and has been on therapy now for over 86 weeks. Her BMI here started at about 50. She is currently at a BMI below 30, and I'll show you some of the data with regards to the normalization of her insulin sensitivity. And I'm not going to show you the data, but she had significant improvements in diastolic and systolic blood pressures, as well as in heart rate, down down by about 8 to 10 millimeters mercury for both, and heart rate down as well. But the impact here is, of course, like at hunger scale, significantly reduced and really steady weight loss, attaining potentially a new equilibrium at later stages, but continuing the weight loss throughout that entire time frame, and she is still losing weight. This is the oral glucose tolerance test in this patient, where you can see here before the study at 13 weeks and in the extension phase at a later stage, where you can see significant insulin resistance and normalization at the later stages, and of course the blood glucose, as you can see here, impacted as a result of that insulin resistance. Second patient, significant weight loss, again, same paradigm, 40.6 kilogram of weight loss here at the end of about 43 weeks. There's about 26% weight loss. Again, she also had a high BMI, started at a BMI of 55, and again, same paradigm, and no significant adverse effects noted in either of these patients. Skin tanning is observed, but there were no other significant effects. And we've learned significantly from both of these patients as a result of the treatment and the long-term treatment that they have now been exposed to. Now, in closing, like I indicated earlier, POMC neurons, potential for Prader-Willi syndrome patients, PCSK1 deficiency present a pathway. We know in wild-type patients that we can get significant weight loss, even in those that are heterozygous for a loss of the MC4 receptor. But it's clear that if you go upstream, that you have more profound patient benefit with regard to the morbid obesity that these patients suffer from. However, there are a large number of other genes upstream where we can look at other modulators, not just missense, nonsense mutations or insertions that defect these genes or imprinting defects, with regards to methylation of the POMC gene, serotonergic leptin receptor effects, nuclear receptors, and a range of others, where we believe we have a large patient population that we are now one by one pursuing to determine what the benefit is of setmelanotide treatment in these populations. We generally try to proceed with rodent studies to identify and prioritize from among this wealth of treatment opportunities which patients will benefit most, and with that then we can make next steps. And hence we are now engaged in a number of trials to look at selected upstream parameters and thereby evaluate the clinical benefits of setmelanotide treatment in severe monogenic forms of obesity. And then in closing, the people that have contributed out of Rhythm, our scientific advisory board, investors, and a large array of collaborators. We are a virtual company, and hence we heavily depend on those contractors, collaborators, and people that can work together with us to move setmelanotide forward towards what we anticipate is pivotal trials for its final NDA approval. Thank you.